Okay, welcome to day 3 of distance learning. So, I'll make this one short and sweet. I wanted to make this a 2-part lesson for this format. Normally we would try to do it in a day, but I felt like that would be ambitious for this. So I want to introduce this topic first, and then we'll get into it on Tuesday with the actual activity. I mentioned this in the previous video, if you watched that. So today's going to be background information just to make sure you get it and understand what goes into it. And then Tuesday we're going to do a simulation on the computer where I'll post a video of it because I'll link the website. And I know I keep saying this if you've been watching, but in the past you have to have a certain plugin for the simulation to work. So I'm just going to err on the safe side and do a video of me running through it so you guys have it in case it doesn't work for you. You can go through it on your own if you want to, but you'll have the video and you can just watch me do it. But either way, so today we've been talking about DNA replication and to kind of transition and not wrap this up because we're going to come back to it, but going from replication into transcription and translation, talking about how the DNA is actually used. We're going to look at a procedure that is done in lab settings called PCR. We're going to talk about what it's used for. So first and foremost, I just want to clarify, PCR is not a biological process in that it doesn't happen in your body naturally. Don't confuse this with replication and think that this is something that your body does. This is something that scientists have figured out and do artificially in a lab setting in order to get a certain result. So let's talk about what PCR is. PCR stands for polymerase chain reaction. So if you remember, and even if you don't, if you go back to the PowerPoint from a couple days ago when we were talking about the different enzymes involved in DNA replication, one of the enzymes was polymerase, and polymerase is the enzyme responsible for copying the base pair. So it's the actual enzyme that's responsible for making the new DNA. So if you break this down, polymerase chain reaction means you have a chain reaction, multiple different reactions that are going on involving polymerase, meaning that you are using polymerase to copy lots of DNA very quickly to get a specific fragment. So there's a lot of applications for this. I'll come back to this at the end. Hopefully I won't forget. But we'll talk about why we're doing this in a lab setting later on. But this is the virtual lab we're going to be doing on Tuesday. There's a website that does a really good job of going through it and showing you how it works and talking about the applications of it. It's fairly short, but I didn't want to overload you all in one day. I want you to have the background information first to make sure you understand it and have you guys ask any questions if you have it. And then we'll get into the PCR virtual lab on Tuesday. So 4 things you need for PCR to work. And this is going to be the question for, or these are going to be the questions for today. So make sure you pay attention to this. Pause this video here, go over to the Google form, answer these questions because you will need to know these if you're going to understand the lab on Tuesday. So 4 things you need. First and foremost is the most obvious. You need the DNA strand that you want to copy. You can't copy something without the original version there. If you want to copy notes down, you can't get notes unless I give you the notes or you get the notes from somebody else. So you need the DNA strand that you're looking to copy. You need DNA polymerase. It's in the name, so hopefully you can get that one as well. You need to have something to copy it down with. You might have the set of notes that you want to write down, but if you don't have a pencil or a pen or something to write with, then you can't copy it down. RNA primers. We talked about this when it came to replication. You have to have primers when you copy DNA because the primers will tell you where to start copying. If you don't have a starting point, you can't start. So you have to have primers in there. If there are no primers, then you cannot start copying at all. And then for free nucleotides, this is the one that I think confuses the most people. What this is saying is a nucleotide is basically those A's, G's, T's, and C's. They're the building blocks of DNA. So the analogy I use for this one is it's like building a house. If you want to build a house, you have to have the bricks. The bricks by themselves are not a house, but when you put them together in the right order, you will get a house eventually. So it's the same thing. You've got the A's, T's, G's, and C's floating around in the cell. They aren't DNA yet because they're just base pairs, but if you plug those base pairs into a DNA strand, they will code for something. So you need to have those if you're going to copy them as well. You need the building blocks if you're going to build. So to summarize it, you need a template, you need something to copy that template, you need a starting point to copy it, and you need the building blocks for which to copy it with, if that makes sense. Email me if you have any questions. All right, couple steps. We'll go over this again more on Tuesday, so if you don't understand this now, don't worry about it. I'll ask this on Tuesday again, but today's focus is mostly just going to be on the necessary requirements. But there's basically 3 major steps for PCR, and you just repeat them for as many sequences as you want, so it makes it kind of simple, which is nice. First thing you want to do is you want to denature it. We haven't talked about this term, I don't believe, but denaturation means that you take a protein and you heat it up so that it no longer works to do what you wanted it to do in the first place. So I know I use a lot of analogies, but I feel like it's best to understand especially molecules on a small level. So imagine you've got a key. The key fits a particular lock, and it will only work on that lock. Same thing with proteins. Proteins have a very specific function, and they will only do a specific thing. However, if you heat a key up enough and you heat it up to melting point, the key will melt and it'll lose its shape, and then the key will no longer work. Same idea with proteins. If you heat them up enough to a certain point, they'll start to fall apart, and then the protein won't do what it's supposed to do. So the reason why we do this is we heat it up just enough so that the DNA unzips itself. This way we don't have to use helicase. We can skip that. We don't need that enzyme for this procedure, and this just cuts down on cost and it makes it a little bit simpler to use. It's important that you get the temperature right, however, because if you heat it up too much, then the DNA will completely fall apart. You want to hit that sweet spot where it's just enough that the DNA will break apart in half and unzip, but if you heat it up too much, then the whole thing will just fall apart. Once this is done, once you have the two parts separated, you want to get your primers in there just like you would during regular replication. So the way you do this is you flood the solution with primers. That way, when you're going to cool down the solution in order to try and get them to bind to the DNA, you have a really high chance that instead of the DNA sticking back together, you get a primer in there. So you're going to get your primers to stick, and then once the primers are on there, you can actually start copying, which is the same as what we did with regular replication. So second step, add a bunch of primer, and then you're going to cool it down. That way, once it cools back down and you're not separating the strands anymore, the primers can stick, and you can go ahead and start copying. And then the third step is elongation. Once the primers are on there, we're going to heat it back up to just a little bit warmer in order to get the copying started, because it needs to be at a certain temperature to facilitate copying. So once the primers are on there, you're going to heat back up. This will activate the polymerase. The polymerase will do what it does, where it copies each of the strands, and then you can run through the DNA strand and get the DNA strand that you desire, and you now have two copies of whatever the strand is that you started with. So that's really it. That's really all there is to it. There's just 3 steps. So heat it up to break the DNA apart, cool it down so the primers can stick, heat it back up so it starts copying. And you just do that as many times as you want to get as many DNA fragments as you want, so you can see how this is a fairly simple process. It's pretty simple, and it will only take a few minutes to complete the whole thing. So you can repeat this multiple times to get as much DNA as you want. This video I'm not going to show here because I'll post it on the Google Classroom, so you can watch this if you don't want to watch through this video a million times. They probably summarize it in a much shorter way. So going back to this, why we do this. So what is the point of PCR? What do people use this for in real life? There are several situations in which you would want a lot of DNA very, very quickly. When you are testing DNA, if you were to get samples from any organism, each cell has a very, very, very small amount of DNA, meaning it only has one set of DNA, obviously. And that's very difficult to work with in a lab setting. So when you're looking at DNA, you want to have lots of copies because that makes it really easy to mess with and look at and understand how it works. It's pretty much the case with anything. If you want to study something, it's easier to study it if you have a lot of samples of that thing. So a couple things this is used for. A couple of the biggest things. Crime scenes. If you ever watch mostly it happens in like the stage CSI kind of shows where it's all played up, but it's based on real science for the most part. It depends on the show. But crime scenes, when they go into a crime scene and they collect a DNA sample and they're trying to trace it back to an individual person, this is how they do it. If you're getting a sample of DNA from the crime scene, you're already not getting a lot to begin with, because it's not like it depends on the crime, I guess, but it's not like they got DNA all over the apartment. You're probably getting it from a very small sample of blood droplets or skin or hair or whatever may be left over. So you're already limited with a very small amount to work with. So you want to make sure that you get multiple copies of that DNA so you can accurately get an idea of who this person was at the crime scene. So what they do is they take those samples from the crime scene, they run it through PCR to get as many copies of DNA as they need, and then they run it through certain tests to see whose DNA this is and who we can go potentially arrest depending on the circumstances and all that. Other things that it's used for: paternity tests. If you guys ever watch Maury or Jerry Springer or things like that, and they do like the whole "you are the father, you are not the father" thing, that's how they do that. You take DNA from the parents, you run it through a test, and you look at it. And to sum it up pretty simply, we'll talk about this more. I've done this in Bio 2, but I don't think we'll do it in Bio 1 this year. But to simply put it, your DNA, since it's unique, will produce a certain pattern when you run it through a certain test. And since your DNA is a mix of your mom and dad's DNA, you will have half of your mom's patterns and half of your dad's patterns. So that's how they figure out if a person is related to another person. If your DNA patterns line up with your dad's or your mom's, then you know who's your parent. And that's how they say, you are the father, you are not the father. Last example I'll talk about, genetic testing, when we're looking at certain genes and how certain genes work. This is how we've been able to identify certain genes in the human genome. So we know, like, for example, this stretch of DNA codes for eye color, or this stretch of DNA is related to cancer-causing genes. This is how we've been able to figure that out, is because when we take those genes and we remove them and we look at them in isolation, we can see what effects they have on the body when they are active or inactive, and we can see what changes occur. But anyway, so that's a very general overview of this process. Go back through this or rewatch not only this video but the YouTube video if it makes more sense to get a basic understanding. Like I said, it can be kind of confusing to start off with, which is why I wanted to break this up into two parts, especially since we're restricted to distance learning. So just for today, just focus on this part here. I always point to the screen as if you guys can see me. Focus on this part here. This is the main thing for today, because this is going to be really important for the virtual lab. They're going to have you, and by you I mean me, because I'm going to be doing it on video, but they're going to have us actually put together the solution with these things in it. So you're going to have to keep track of what these things are and what they're for. So focus on that today, and then on Tuesday I will ask you more about these steps. What is the point of denaturation? What's the point of annealing? What's the point of elongation and all that stuff? So make sure you understand this. Email me if you have any questions. Watch this video if you need more information, and just be ready for Tuesday, where I will give you another set of questions based off of this. So have a good weekend. Hopefully this first week of distance learning went fine. Didn't have any major problems. I think you guys did a pretty good job for the most part. Good job this week. Keep up the good work. Got a couple more weeks of this, and let me know if you have any questions. So have a good weekend.